thank you for the opportunity to present my research, which, as Bill said, is the idea that there's a genetic mechanistic connection between the processes governing aging or increasing age or longevity, whatever you'd like to call it, and the same processes that play a role in carcinogenesis. And one of the arguments I'll make, and really this is the first time I've had the opportunity to take this work out and present it because it was recently published in October, the idea that this family of aging genes referred to as Sirtuins or Sirt genes, that Sirt2, which is the cytoplasmic Sirtuin, is a pancreatic tumor suppressor, Sirtuin. And I stole that title from one of the reviewers who wrote a preview for our manuscript that was just published. So in my laboratory, this is really the idea of what we work on. And that's the idea of what is translational research. And even though we talk a great deal about what translational research is, I think that it's somehow a little more difficult to grab a hold of it other than say we're working on translational research. So for me, translational research is a clinical result or a clinical observation. And what we do is we use that clinical observation to make a basic science observation. And I'll talk a little bit about that today. And then, of course, once you have a basic science observation like a mouse model, you can have preclinical results, preclinical experiments, and then, of course, you can look at human samples. And then, of course, you use all of these results to go back to where you started. That is, maybe do a clinical trial or go back and look at clinical results. And it's kind of a never-ending direction. And in our lab, we like to follow what is Harold's provocative question number 7, which is how does lifespan of an organism affect the molecular mechanisms of cancer development? And more importantly, can we use these to I'm not sure deep in our knowledge, but can we use these as ways of developing chemopreventive or anti-cancer or cancer recurrent preventative agents? So what's longevity? I mean, we talk a lot about aging. I mean, each of us looks older than we were 10 years ago, but I still think the ideas of aging and longevity are somewhat ethereal. And this is a slide I grabbed off of a blog about 10 years ago, and I still almost show it in every talk I give, and I think it's quite enlightening. If you look at survival in Rome, it wasn't very good. And my guess is that for many reasons, once you were born, you went down a slippery slope, and your average lifespan was quite low. But right about 200 years ago, if you look at the curves for longevity, a very interesting thing occurred. You have the same curve as you did 1,800 years ago, but suddenly there's an inflection point, okay? And then you're on a different curve. It's not a great curve, but it's a change in curve. And if you look at survival today, it's not quite this straight. But it kind of goes down a little bit. But it's relatively straight. And this is true for either survival or curve D is if you remove cause-specific survival like cancer or heart disease, heart attacks, things like that from eating meat. And what you see is longevity is two very separate slopes. It's a very relatively flat slope. So as long as you don't get hit by a car, you're going to survive. And then there's an inflection point, the same inflection point we saw 200 years ago. And then once you fall off of that curve, that's it. A species can no longer support longevity. And if you really look at this curve, it suggests to me that there must be some genetic or signaling processes that maintain this very flat curve. And at some point during your longevity span, these processes no longer exist, and suddenly you fall off the survival curve. And while there is a relative variability of how long you may survive, it's downhill from there. So what we really want to bring out in this slide is this inflection point. And the reason this is so key is because I'm going to make the argument that Sirtuins play a role in that inflection point. And if Sirtuins play a role in that inflection point, maybe Sirtuins play a role in carcinogenesis or preventing carcinogenesis. So what are Sirtuin genes? Well, they were isolated in yeast and C. elegans Drosophila. They're referred to as silent information regulator proteins, Sirt2 genes. And that's in yeast. In humans, they're called Sirtuins or Sirts. So Sir genes in yeast, Sirt genes in humans. And the interesting thing about them is they seem to respond to respiration. They're regulated by the NAD, NADH levels. These are packets of the cell that determine energy levels. And if you ask me, the most important thing is the Sirtuin genes appear to be turned on by caloric restriction. Okay? And caloric restriction not only turns on Sirtuins, it's a well-known observation that caloric restriction prevents cancer, as well as other diseases of aging, like neurodegenerative diseases, insulin resistance, and whatnot. And actually, the reason I really bring this out is this slide. Okay? This is how the Sirtuin genes play a role in regulating longevity. Okay, so if you look at C. elegans, and the average C. elegans, which is a little nematode, it's a little worm, they live about 3 weeks. Okay, and interestingly, they have a very similar survival curve as mammals. It has a very flat slope, there's an inflection point, and then after that, the worm can no longer exist. Now, if you take the Sirtuin genes in C. elegans, and you overexpress them, so you knock in 3 of the Sirtuin genes, what happens is the longevity of that species increases. It increases from 3 weeks to 5 weeks. But the interesting observation is here. The inflection point doesn't change. And once you hit the inflection point, that secondary curve doesn't change. What changes is the length of the primary slope. Is if Sirtuin genes play a role in regulating whatever processes play a role in keeping this slope very straight. And if you do the opposite experiment, that is, you take the Sirtuin genes in C. elegans and you remove them, what you see is the opposite result. You can see that there's still an inflection point. Once you hit that inflection point, that's it. You're off the survival curve. What changes is the relative length of the initial slope. To me, suggesting that what Sirtuins do is they regulate the processes governing longevity in maintaining that ability of a species or organism to survive. And I want to go back real quick and talk about what Sirtuins do. Sirtuins are post-translational modifying proteins. We know that one of the major signaling pathways in the cell is via kinases, or the kinome. A protein comes along, it gets activated, it's a kinase, it finds its downstream target, it phosphorylates that protein, and that protein changes in function. So that protein goes from on and off because a phosphate molecule has been put out. And then a phosphatase, a protein that takes that phosphate molecule off, takes it off, and it changes that protein back. So what do Sirtuins do? Well, Sirtuins don't play a role in phosphorylation. They play a role in a post-translational modification referred to as acetylation. And it's very similar. It's a lysine amino acid gets acetylated, and much like a phosphorylation, it's a post-translational modification which regulates the activity of the protein. What Sirtuins do would be the acetylome version of a phosphatase. They don't take they don't put an acetyl molecule on a protein, they take it off. Much like a phosphatase takes a phosphate molecule off a protein. So this really sums it up right here. You have a protein, it's inactive, it has a lysine and it's acetylated. A Sirtuin molecule comes along, you get a protein-protein interaction, requires NAD as I mentioned, and suddenly you have a deacetylation. This protein now lacks a lysine that's acetylated, and the protein goes from an inactive form to an active form. So they're acetylome version of a phosphatase. Okay, let me just jump ahead here. And in humans, it turns out there's 7. 3 in the nucleus, 3 in the mitochondria, and 1 in the cytoplasm. I'm not going to talk about these 2, but we've published papers on these in the past, a couple of these in 2010, and the manuscript showing that Sirt2 is a tumor suppressor gene for which these results I'm presenting today was published in October. So Sirtuin is the molecule in the cytoplasm that's turned on by caloric restriction that, at least in primitive organisms, plays a role in longevity, and that is it regulates that long portion of the curve before it hits the inflection point. And what do Sirtuins do, just to very briefly? Well, 3 are in the nucleus. Sirt1 plays a role in nutrient A availability, cell stress, and survival. Sirt6 plays a role in DNA repair. There are 3 Sirtuin molecules. They're in the mitochondria, and as you might imagine, the mitochondria makes energy, so these Sirtuins play a role in connecting caloric restriction to energy generation and control. And what I'm going to talk about today is Sirt2, and Sirt2 is the only and primary cytoplasmic protein deacetylase. Okay, so why do we work on Sirtuins? Okay, well, 7 years ago when I was still at the NCI before I went to Vanderbilt, the Sirtuin molecules had just been discovered by several investigators, including Lenny Garante and David Sinclair at MIT and Cynthia Kenyon at UCSF. And of course, we were interested in the idea because these proteins play a role in longevity. And this is really the curve that caught our interest. This is incidence of cancer as a function of age for solid tumors. So this is breast, prostate, pancreas, lung, sarcomas. And the interesting observation is, and this is well known, at least for solid tumors, not for pediatric tumors or reticular endothelial tumors like leukemias and lymphomas and whatnot. If you look at solid tumors, you can see the single greatest prognostic factor in developing a tumor is increasing age. And the reality is, cancer is primarily a disease of the elderly. And if you look as a cancer incidence as a function of age, even though the cell deaths, it should say incidence, what you see is it's a relatively flat curve until you get into your 50s, and suddenly there's an inflection point. And the percentages of getting a cancer go up very significantly. And one of the reasons we work on Sirtuins in cancer is when you take these two curves and you put them together, this is what you see. You see Sirtuins regulate longevity. There's an inflection point, and they can no longer regulate longevity. If you look at cancer incidence, you see there's a relatively flat curve. There's an inflection point. So 7 years ago, because we had the resources available from the intramural program, we made a very simple and I thought logical hypothesis. The aging is the most important prognostic variable in cancer. The Sirtuin proteins play a role in regulating longevity or aging. Hence, would Sirtuin proteins play a role as tumor suppressor genes? And of course, we know there's no such thing as a tumor suppressor gene. The cell hasn't evolved to have proteins to prevent cancer. What these are repair proteins. They make sure the cell does the things that it's supposed to do. And if it's gone, or its activity decreases with increasing age, well, then the cell is at risk for something bad to happen. And that can be anything: neurodegenerative disease, insulin resistance, including but not limited to carcinogenesis. So 7 years ago, we knocked out 3 of the primary Sirtuins: Sirt1, Sirt2, and Sirt3. And today I'm going to talk a little bit about Sirt2, the cytoplasmic tumor suppressor gene. So what is a tumor suppressor gene? Well, this is tissue, have a tissue culture phenotype consistent with the 2-hit tissue model for immortalization, and I'll talk about that in a couple slides. So it is a tumor suppressor gene in tissue culture in the little incubators that we keep in our laboratories. If we make a mouse lacking this tumor suppressor gene, they should get tumors. And the tumors they get should give us insight into what kind of role these proteins might play a role in human malignancies. And that's if they get tumors. And of course, we hope they will when we made these mice, and we were lucky that they were. And then, of course, you should see a loss of function, or maybe more importantly, decreased protein levels in human tumor samples, much like I showed in my first slide. Right? You have a clinical observation, you make a mouse for a basic science observation, you do preclinical work, and then you look at human samples. So you should have altered protein levels in human samples of human malignancies, and these should match the tumors that you get in mice. So if the mice get breast or pancreas cancers, we should see that these proteins are decreased in human samples of patients who have breast or pancreas cancer or lung or liver or whatever we're working on. Okay, so I'm going to show a few experiments to prove to you that Sirt2 is a tumor suppressor, certainty, and then I'm going to show a few slides to show that it plays a role in pancreas cancer, and then a few slides suggesting there's a connection between Sirt2 and KRAS, which we know is a very important protein in GI malignancies. So this is the experiments for which Harold Varmus and Michael Bishop got the Nobel Prize. What they showed is you could take primary cells, non-transformed, normal tissue culture cells, and if you infect them with 2 oncogenes, they go from a hyperplastic to a malignant tissue culture phenotype. And this is a very simple idea. A normal cell goes to a transformed cell. It needs a series of events, an initiating event, and then a series of pro-proliferative events. And in tissue culture, you can do this with 2 oncogenes referred to as MYC and RAS. And several years later, several investigators, including Robert Weinberg and Bert Vogelstein and whatnot, they showed that you can substitute one of these 2 oncogenes with a loss of a gene. And if this loss of gene complemented one of the 2 oncogenes, we refer to them as a tumor suppressor gene. And that's really the idea behind tumor suppressor genes. So the question is, if you take cells lacking a gene, can they be immortalized or transformed by a single oncogene? And that's shown right here. So the first thing we did is, after we made our knockout mice, we made tissue culture cells, which we referred to as mouse embryo fibroblasts. We took the wild type cells, which are plus here, Sirt2 plus, and the knockouts, which are Sirt2 minus. And what we did is we infected these cells with a virus that expressed either the MYC oncogene, discovered by Harold and Mike Bishop, or RAS, or both. And this is the experiment that people had seen for many years. One oncogene does not immortalize or transform primary mouse embryo fibroblasts. You need both. But if you take cells lacking Sirt2, they can be immortalized by a single oncogene. And by definition, that suggests that these proteins are tumor suppressor genes. And there are, of course, other experiments are required to make such a large and significant statement, but I don't have time to show those. But you can look at the manuscript, and it's in the supplement or in the manuscript itself. So Sirt2 is a tissue culture tumor suppressor gene. Well, okay, what about in vivo? Because the fact of the matter is a mouse and a human act a lot differently than tissue culture cells in an incubator, which is unfortunate for us that work in a lab. So what did we do? We took mice that either were wild type for Sirt2 or knockout, and we saw whether they would get tumor genesis spontaneously in knockout mice. And what we saw is both the female and the male mice get tumors. The female mice get tumors starting at about 10 months, and the male mice get tumors starting a little bit sooner. And if you look at the tumors in general, you can see this is a pretty strong tumor suppressor gene. A significant amount of breast tumors, and then the rest of the tumors that the mice get, for the most part, are tumors associated with GI malignancies. And if you go on and look at just the females, which I'll talk about just for a second, about 90-some% of female mice all get mammary tumors. So the Sirt2 knockout mouse is a mouse for breast cancer, and not any kind of breast cancer. It's a model for receptor-positive luminal B breast cancer, which of course is interesting. And if you look at these mice, almost all the female mice get breast cancers. But more interestingly, if you look at the male mice, what you find is a very different observation. Male mice, none of these mice get mammary tumors. And if you look at the female mice, outside of liver tumors, they only get breast tumors. But if you look at the male mice, you can see the male mice overwhelmingly get GI malignancies. So these results suggest several very interesting results, right? This suggests that an aging gene is a tumor suppressor gene, that the knockout mice not only get tumors, but they get gender-specific tumors. That is, the tumors in female mice are significantly and completely different than the tumors that you get in males, suggesting that in some way the Y chromosome, which affects intelligence because women are smarter, obviously also affects the kind of tumors that these mice get, which is really a staggering observation. But to me, the really interesting observation is not just that about 20% of these mice get pancreas tumors, but except for pancreas cancers, every tumor that these mice get are associated with GI malignancies. Okay, so why is this important? Well, of course, as I showed on one of my first slides, it's not just enough to have tissue culture data and an observation based on clinical data, that is, there's a connection between aging and cancer. That is, you have to look at human samples. Because if the human sample results don't match what you get in mice, I mean, it doesn't mean you shouldn't study it, but it gives it more clinical, physiological relevance. So what we did is we looked at expression of Sirt2 in human pancreas tumors. We did this using genomic data from the Oncomine repository archive at the University of Michigan, and then we did tissue array data. And what these are pancreas tumors that are cut and put on slides, and you can buy them from commercial companies, and you can stain them with your antibodies. And what we found is looking at 3 genomics, so this is RNA expression, looking at 3 genomic studies that we pilfered from University of Michigan archive, you can see to relatively high p-values, Sirt2 expression is decreased in normal pancreas versus pancreas adenocarcinoma. And when we bought a commercial sample, you can see the ductal cells in normal pancreas express relatively high levels of the Sirt2 protein. But if you look at tumor cells, you can see very little protein is regulated, suggesting but not proving that Sirt2 may play a role, perhaps an early event role, in carcinogenesis and GI malignancies, including but not limited to pancreas tumors. Though pancreas and liver are the most common malignancies that we see in these knockout mice, at least in the male knockout mice. Okay, so why am I here? Well, one of the first things you get to know when you make a new mouse and you get a pancreas tumors and you call the pancreas scientist at your university, which is Vanderbilt, and you show your results to them, the first thing they say to you is, well, have you looked at KRAS? And since I hadn't really thought of that yet, that's of course the first thing we did. So one of the first things that was suggested to us, and the first thing we did was, is it possible that Sirt2 interacts with KRAS and it regulates the normal enzymatic function of the KRAS protein? And of course, when you have a correct interaction between Sirt2 and KRAS, you should have normal cellular function. So the hypothesis was a very simple one, That is, if you don't have Sirt2, there's no protein to interact with KRAS. You can have aberrant enzymatic activity, and instead of normal cellular function, you could have increased cellular division and carcinogenesis. So very, very simple and obvious hypothesis, yet one that very much fits the data that we had previously published. And the rest of the data I'm going to show has not been published yet. And if you're watching this, please don't beat us in the publication. So the first thing to do is simply, is there a physical interaction between the Sirt2 anti-aging cytoplasmic Sirtuin protein and KRAS? And in fact, there turns out to be. And to do this, we did a series of what we refer to as transient transfection experiments. We take the KRAS protein that has a little HA tag on it. That's a way we can immunoprecipitate it with an antibody. And we take a Sirt2 protein, which is flag tagged, and we just transfect them into cells. We harvest those cells and we IP flag, which brings down KRAS, and then you can see KRAS comes down, and then what we do is we take the other half of the sample and we do a Western with the HA antibody. And you can see there's a physical interaction between KRAS and Sirt2. And that's true if we do the other experiments, same thing. If you do a Western for flag and a Western for HA, you see the same experiment, there's a physical interaction, and we do it for a third time. So we do this 3 times to definitively show there's a physical interaction between these proteins. And while this suggests that there's a regulatory interaction, it doesn't prove it. It just suggests that they interact. Well, what about activity? Well, we can do activity assays. And to do this, we took wild-type mouse embryo fibroblasts that I talked about in our first set of slides, and in knockouts, and what we see is here's the wild type. Here's an experiment that looks at enzymatic activity of KRAS. And you can see that the wild type cells, there's significant KRAS activity and it's decreased in the knockout cells. And if you do a similar experiment, not with wild type and knockout cells, but doing shRNA, which indirectly removes Sirt2 RNA and protein from the cell, we see the same result. There's a decrease in KRAS activity when Sirt2 is gone, and we did this in 2 separate experiments. Clearly showing not only that Sirt2 binds to KRAS, but it regulates enzymatic activity, suggesting perhaps that Sirt2 might be an early event in pancreas and other GI malignancies. And when Sirt2 activity goes down with age, which is the supposed mechanism of how they work, that puts the cell at risk because there's the aberrant regulation of KRAS activity. Okay. If that's the case, then you would think that KRAS would be deacetylated by Sirt2, because that's what Sirtuins do, right? Sirtuins are the acetylome version of a phosphatase. They don't take a phosphate off a protein, they take an acetyl group. So the next thing we did is we saw if these experiments are correct, and these are relatively new. So what we did is we took KRAS protein that we immunoprecipitated out of a cell, and the cell was arranged so that it has very little deacetylation activity. So the protein we're IPing is hyperacetylated. It has as many acetylated lysines as possible. And then we take that and we add the Sirt2 protein, and what we see is without Sirt2, this is KRAS, and it's relatively more acetylated. When we add Sirt2, it becomes deacetylated. Okay? Suggesting that not only does Sirt2 physically bind to KRAS, but it deacetylates KRAS as well, suggesting there probably is a role in regulating enzymatic activity, and it's a direct effect. So the next thing we did is we took this protein, and we just did this experiment a couple weeks ago. We ran the protein extracts on a cell. We isolated KRAS. We sent it for mass spec, which of course is necessary for us to determine what the specific lysine is. And that's just right here, as I was saying before, right? You have a protein, KRAS, it has a lysine molecule. You add Sirt2, it becomes deacetylated. You can take these 2 samples, the inactive protein and the active protein. You can send it for mass spec, and it'll tell you what the specific amino acid is, which is deacetylated. And this is the one we found, lysine 117. And it turns out lysine 117 resides in the conserved guanine nucleotide binding domain of KRAS. Can everyone see that right there? That's the lysine right there, 117, that it's right next to the catalytic domain of the protein in a perfect location for acetylation and deacetylation by Sirt3 to regulate enzymatic activity. So I'm almost over. I'm going to go very quickly and just show one more slide. Because I've shown that they they regulate each other, but that's not physiologically important because one protein deacetylates another protein and they physically interact. That could have nothing to do with carcinogenesis. So we definitely need to show a few experiments suggesting that the aberrant regulation of KRAS plays a role in its ability to cause cancer, at least in tissue culture. And that's shown right here. Here's the wild type Sirt2 mouse embryo fibroblast, and what we did is we transfected it in the viral pro-oncogene of RAS. Not exactly the same as KRAS, but it's a reasonable place to start, at least until we have more reagents and we can do more elegant experiments. And you can see if you do a colony formation assay, which is an indirect way to look at the ability of a gene to cause cancer in tissue culture, you can see you get a couple colonies. But if you put these RAS viral oncogene in the knockout cells, you can see there's a huge increase in the ability to cause transformation and immortalization in tissue culture. And this is just another experiment shown right here. And I have one more. Well, I'm going to skip this. It's the same idea, right? If you look at wild type RAS or knockout with RAS, this is just another experiment. You can see that RAS has a much stronger ability to transform cells lacking Sirt2 than it does wild type cells, suggesting not only that Sirt2 binds KRAS, it deacetylates KRAS, it regulates enzymatic activity, but loss of Sirt2 also allows RAS to play a role, to elicit a stronger pro-carcinogenic phenotype when there's both aberrant regulation of RAS and loss or aberrant regulation of Sirt2, suggesting they might be connected genetically in some way. So what are we doing now? Well, thanks to a pilot grant, what we're doing is we're taking the KRAS mouse and we're crossing it with Sirt2 so we can have a conditional KRAS mouse and we can have a mouse with either wild type Sirt2 or a mouse that lacks the Sirt2 gene. So what we're going to try to do in mice, what we've done in tissue culture, to see if we can develop a model for the development of pancreas cancer in mice, right? Much like the slide I showed, right? Clinical observation, right? An in vivo mouse model. And then we would use that mouse model to do preclinical data, look at human samples, and then think of ways to do chemoprevention and anti-cancer. So that's where we are right now. We don't really have these mice yet. We have these mice, but we have to cross them with a special secondary mouse to knock out Sirt2 only in pancreas ductal cells. So what we're going to have is a mouse that expresses KRAS in pancreas ductal cells, and these mice also lack Sirt2 in those cells, and we're going to see if we can elicit pancreas tumorigenesis. And I don't really have time to go into kind of the nuances of what we think the mechanism is, but if you want to come up, I'll discuss it with you. But it plays a role in senescence. And Sirt2 is a tumor suppressor gene, connects aging and cancer. The knockout mice develop a series of tumors, gender-specific tumors, and mostly GI tumors in the male mice, and they're all adenocarcinomas, ductal adenocarcinomas. And we think this suggests a connection between aberrant metabolism, because they're turned on by caloric restriction, and the underlying mechanism is that Sirtuins may protect against carcinogenesis. And here's my laboratory. As you know, I don't do any of these experiments. I just sit in my office and tell them to do them. So Hai-Yang Yang is my lab manager. Young Hong is a radiation oncology resident who works in my lab. Hong Kim is my senior scientist who just got a faculty position in Korea, so unfortunately I'll be losing him. And these are the rest of my postdocs, and they all do a wonderful job, and they're really serious, dedicated, and rigorous trainee scientists. Thank you.